Hello everyone, welcome back to our microbiology class. Today's session is on chapter 12 from the book, and it's on slime molds. The learning objective is to compare and contrast cellular slime molds and plasmodial slime molds. Slime molds are closely related to amoeba and are placed in the phylum of Amoebosa. There are two taxa of slime molds, cellular and plasmodial. So the cellular slime molds are typical eukaryotic cells that resemble amoeba. In life cycle of cellular slime molds, as you can see in the picture, in the figure 12.22, the amoeboid cells, they live and grow by digesting fungi and bacteria by phagocytosis. So the cellular slime molds are of interest to biologists who study cellular migration and aggregation, because when conditions are unfavorable, large number of amoeboid cells, they aggregate to form a single structure. This aggregation occurs because some individual amoeba produce the chemical cycle EAMP, or in other words, we call it as CHEM. So towards which the other amoeba migrate. Some of the amoeboid cells form a stalk. Others swarm up the stalk to form a spore cap. And most of these differentiate into spores. So when spores are released under favorable conditions, they germinate to form a single amoeba. In late 1970s or early 1970s, a Dallas scientist discovered a pulsating red blob in his backyard. And the news media at that time, they claimed that the new form of life had been found. For some people, this type of creature which was found evoked a spine-chilling recollections of an old science fiction movie. Before migrations got carried away too far, the scientists, they calmed everyone's worst fear that by identifying this amorphous mess as merely a plasmodial slime mold, but its unusually large size, which was 46 centimeters in diameter, and even the scientists got startled. Plasmodial slime molds were first scientifically reported in 1729. A plasmodial slime mold exists as a mass of protoplasm with many nuclei. It is multinucleated, and this mass of protoplasm is called plasmodium. The entire plasmodium moves as a giant amoeba. It engulfs organic debris and bacteria. The bacteriologists and biologists in general have found that the muscle-like proteins forming microfilaments account for the movement of the plasmodium. When the plasmodium slime molds are grown in laboratories, a phenomenon called cytoplasmic streaming is observed, during which the protoplasm within the plasmodium moves and changes both its speed and direction, so the oxygen and nutrients are evenly distributed. The plasmodium continues to grow as long as there is enough food and moisture for it to thrive. So when either in short supply, the plasmodium separates into many groups of protoplasm. Each of these groups form a stalk sporangium, in which haploid spores, which is a resistant resting form of the slime mold, develops. When conditions improve, these spores germinate, they fuse to form a diploid cells, and develop into multinucleated plasmodium. And finally, with this, we have finished today's lecture on slime mold. And I thank you very much to everyone for watching, and I hope everyone enjoyed it. Thank you.